My name is Bakish Hamina binti Adam, 97298 from Resource of Biotechnology.
I am Magdalene Tenkawe. My metric number is 97500 and I'm from Resource Biotechnology.
My name is Dania Karenisa binti Madani, 97367. I'm from Resource Biotechnology program.
My name is Valerie Coalition, 97858 metric number. I'm from Resource Biotechnology.
I'm Greta Yo Yu Qian, 97428 metric number, and I'm from Resource Biotechnology.
Introduction. In biochemistry, 3,5-dinitro-salicylic acid (DNS) is widely used to estimate reducing sugars. Several reagents have been used to measure sugars by taking advantage of their ability to reduce. This technique uses reducing sugars of free carbonyl groups, CO. This entails the oxidation of ketone functional group in fructose and the aldehyde functional group found in glucose. Under alkaline circumstances, 3,5-dinitrosalicylic acid (DNS) is simultaneously converted to 3-amino-5-nitrosalicylic acid. The DNS method is based on the reaction between reducing sugars and the DNS region, resulting in the formation of a colored product. The intensity of the color is directly proportional to the concentration of reducing sugars present in the sample. This principle forms the basis of constructing a standard curve, allowing for the accurate quantification of sugar concentrations.
Material used in practical 3 are spectrophotometer, hot plate, glass beakers, cuvettes, test tubes and test tube rack, micropipettes and tips, distilled water, standard glucose solution, sodium-potassium tartrate, and dinitrocyclic reagent.
Methods. First, the following reagents are pipetted in duplicate into a series of dry clean, and test tubes are labeled as indicated in the following table. Second, after replacing the above-mentioned solutions in the labeled tubes, they were shaked well and then were placed in a boiling water bath for 5 to 7 minutes. Third, the tubes were cooled, then 1 ml of Rochelle salt was added, then the solution was transferred into cuvette. The extinction of the colored solutions were read at 575 nanometer using test tube 1 as a blank.
In conclusion, this experiment exposes us to the 3,5-dinitrosalicylic acid or DNS method to quantify sugar. The concentration of sugar can be justified by measuring the absorbance with a spectrophotometer. The proper calibration and use of spectrophotometer is practiced to measure the absorbance. Next, the concentration of reducing sugar is determined using the DNS method by determining the absorbance spectrum of glucose at differentiation concentration using spectrophotometer. Calibration curves of calorimetry is graphed to present the linear relationship between the concentration of sugar and absorbance. The use of spectrophotometer to prepare glucose standard curve is being discussed and understood.
For practical 3, the determination of sugar using the DNS method was a new knowledge for us. The process involved carefully preparing the samples, following the protocol for the DNS method, and using a spectrophotometer to measure absorbance. Getting an early exposure of this method helps us to understand the DNS method in detail and its applications in biochemistry. Each steps are crucial in order to prevent any mistakes in the data. As for the difficulties, first, the micropipette that we used for the whole session were contaminated with a yellow solution, which can lead to inaccuracy of the data for spectrophotometry. Thus, we use other groups' micropipette to prevent this situation from happening. Second, misread the manuals also will affect the whole experiment, and we might need to repeat it again.